Good afternoon, everyone. My name is Dr. Gaetan Burgio from the Curtin School of Medical Research at the Australian National University, and I would like to give an overview on an important matter, and I titled the presentation, Any Idiot Can Do It, how easy knockout and knock-in mouse could be generated using CRISPR-Cas9 gene editing technology. So before starting with an important issue, which is replications in the gene editing field, I would like to give an overview what my laboratory is doing. We are mainly a microbiology laboratory, and we are in the interface between microbiology and gene editing technology, genome engineering. So what our core interest is on exploring bacteria and archaea genomes. And what the particular interest is on discovering new antiviral plasmid defense system in bacteria. And we are interested in three main systems, so prokaryote argonauts, CRISPR-Cas systems, and other mechanisms of defense. And from there, we harness the gene editing technology to edit mouse, cells, and applications such as gene therapy. So for this topic today, we focus more on the mouse and the cells. So replication is an important, so we have a replication crisis in science. As you can see in the screen, there are a couple of papers outlining some important problem in science, which is the lack of replication studies and the lack of reliability of the result in science, to a point that recently a paper described this as the natural selection of bad science. So the question is what we can do about it, what is about. And there are three factors that are outlined there. The first one is there are tons of underpowered studies. We are facing the publish or perish system that disadvantages meticulous science, and the structure of the current scholarly publication and funding model disadvantaged meticulous science. So so let's take an example. If you PubMed search improve and CRISPR, you will find 612 hits. And to give you an idea, 1 or 2 years ago, it was only 200. So a lot of colleagues are interested in improving CRISPR. And one example there was, for example, a way to improve gene editing technology is to use what we call non-homologous end joining inhibitors. What this molecule does, so like SCR7, is to to improve the generation of point mutation, for example, in mouse. So how it works, we deliver the Cas9 protein or the Cas9 mRNA with SCR7, we inject into the mouse zygote, and what we noticed, and you can see in the columns labeled HDR, is to increase efficiency in generating point mutation in mouse and in cell line. The problem was, and if you look at the red rectangle, this was only done in a handful of embryos, not a lot. And we were all enthusiastic on this technique, and what we try to do is to replicate the study. Whereas it does work, SCR7 definitively improves HDR efficiency, the problem was it kills as well a lot of embryos. Therefore, we were not able to widely implement the technique. Some people are still doing, I think it's valid. However, there are some drawbacks in using that technique. And so the question is, so what we make of the studies, how to improve reproducibility in science, and especially in the gene editing field. And I give you two examples there that really illustrate how we can do as a community to improve the reliability of the result in the gene editing field. The first one is quite old example, but really telling, is the use of an Argonaut protein called NgAgo. And the second one that we have recently published on the use of a method called two single-strand NgAgo, two single guide RNA system for generating conditional knockout. To give an overview what are prokaryote Argonauts, these are a defense system mechanism against plasmid and viruses in bacteria and archaea. There are a wide variety of defense, could be DNA or RNA guided. As you can see in the cartoon, it degrades the plasmid and the genome. It's a highly diverse and fascinating protein family. What was the story about? It was the use of DNA-guided NgAgo, so Natronobacterium gregory, to edit cell lines. And what was the claim of this paper? One, firstly, the Argonauts are capable to edit at 37 degrees and to generate double-stranded break. The second claim was the editing is almost identical to the CRISPR-Cas system. And if you look at the figure, at the cartoon, so the A, B, and C, you will see that it was used to replace a chunk of the genome. So we were very enthusiastic about this paper, and we all tried. Unfortunately, it doesn't work out that well, and I started to raise some concern about this paper a month after the publication. So 2 months after the publication, so keep in mind that the publication was in May 2016, and early August 2016, I raised some concern about the technique, saying that we have a problem there, it doesn't work as it was advertised. And indeed, it was widely shared, and that with 15,000 reads in 2 days, and that gives you an idea how powerful this post, to my surprise, was in raising concern about the technique, and how quick it was, because it was only 2 months after the paper was published. And what happened next is many laboratories tried to replicate the original studies and start to publish months after the paper was published. So with, for example, those publications there, and there. And we published indeed a replication study a year after, so quite a long time. And what we found is, in short, the protein is well expressed in mammalian cells, so the protein is there, no doubt about that, it doesn't degrade, and secondly, it doesn't edit. So we tried in different systems, in cells, in mouse, and we couldn't find any editing, whereas we could find editing pretty easily with CRISPR systems. So what is the current hypothesis of the use of the Argonauts? Is, in fact, the NgAgo enzyme doesn't edit, but it enhances homologous recombination in bacteria. How it interacts with an enzyme that is important for DNA repair in bacteria called REC-A. So what happened to the paper? It was retracted a year later. And the take-home message from this is we validate or not a study by the accumulation of data and replication studies from multiple laboratories. It doesn't come for a single laboratory. This is really critical. And so the question is, okay, so we need replication, we need multiple laboratories, but what we can do as a community. And I really would like to emphasize this example on this technique. And the story came from this new story in Science that was published, I don't know, something like two years ago, on this quite true and provocative statement that is part of my title, is any idiot can do it, and that gives you an idea how fast the technology has moved to a point now that we can claim that everyone can do it. And that was the substance of this claim. So what we did, we were a little bit cheeky, is to check if it is indeed true. So I explain what the story is about. Is when we need to generate a conditional allele, so for example, to knock out a gene but at specific time and specific tissue, what we do with CRISPR technology, we excise the exon. So you see as the red arrows, we use two guides. What happened? We repair using two single-threaded oligo. So that oligos insert into the genome, and we have 2 logs P on the surrounding the exon 2. What happened next? We cross with a cray, and it excises this second exon. So this landmark study was published in 2013 in Cell, and it uses this technique to insert a gene called MECP2, as you can see in the cartoon A. And the really important feature I would like to point out is the success rate was good. If you look at the table, please ignore the rest, just focus on the red rectangle, and what is pretty clear is the success rate was around 16 to 15%, which was remarkable given that the average success rate to generate a conditional knockout is less than 1% with traditional technique. So we were a lot enthusiastic, and my laboratory started to replicate this exact study. What I did is to use a similar mix, inject into the zygote, and look at the outcome for editing and so on. And what we did is to find that we could insert the LogSP sites, however, we couldn't find the correct insertion. And so there are 3 takes on this. Is one, of course, we might have missed on the specific experimental conditions. Secondly, we lack skills in the laboratory to successfully insert those LogSP sites. And thirdly, it's only on 100 eggs, and the sample size is very low. So what we start to discuss within the community with one of my very good friends, Gaumurthy Shabana Vazaya at the University of Nebraska, we start to discuss why it doesn't work. And what we did is to replicate with 2 other laboratories, so Guru's lab in Nebraska Uni and Petr Kasparek at Prague. And if you look at the table, we had a similar result. We couldn't get a correct insertion of the LogSP. So same again, it could be a lack of sample size, underpowered studies, that makes it difficult to interpret why it doesn't work. So it doesn't tell much. So with Guru, what we decided to do is to survey the community. And what we did, we asked people around in the States, in Canada, in Europe, in UK, in Japan, in Australia, so 20 laboratories worldwide. What is their experience with the technology, how good they were, if they were successful, not successful, and so on. And what we ended up with is we gathered over 50 loci, and what we found is over 18,000 zygote injected and 14,000 transferred with close to 2,000 live pups. Only 16 got the correct insertion for those allele, and only 13 genes were successful out of over 55, which means it requires something like 1,000 eggs to be injected to see the success, which is considerable. And when you take this into perspective, the success rate is equivalent to the classical targeting system, which is giving an idea that it's not as easy as it was advertised. So we published this as a preprint over a year ago, and it was picked up from the news. And when we submitted the paper for peer review, 3 criticism came through, and there are fair criticism. One is they interrogated us for the lack of skills. Secondly, there were differences in mouse strains with the initial study. And the third is the problem with the technique we use to generate those knockout, those conditional allele. So they are all fair criticism, and we try to address those. So the first criticism is the lack of skills. How come we can assess our ability to generate these gene editing mice? One is simple, is to use HDR efficiency, so the generation of point mutation, as a proxy. And what we did is to collect the data on 330 loci, and in terms of data, that was quite considerable because it was on over 64,000 zygotes. And out of the 64,000 zygotes, we had 10,000 live pups, and with 1,369 correct mutation, which correspond to 13%, which is within the average of success for HDR point mutation. And the number of genes successful were close to 300. We were pretty much all successful, which means, as a whole, we do have the skill to generate those single point mutation. However, it doesn't answer the 2 other questions. What about the technique? So the way we address this is, as you can see, the original technique is to generate double-stranded breaks, so 2 cut across the exon, and to replace with the 2 single-stranded oligo. And what we did, we compared with other techniques that use these the vector, so such a DNA plasmid or a long-stranded oligo that replace the entire chunk of DNA. And what we did is to compare side by side for 18 loci. So if I summarize all of this, so we have 4 techniques there. One with the original technique on the bottom side of the table, so the 2 oligo, 2 guide. We did a sequential insertion. So what that means is we do the 2 guide, 2 oligo approach, and only 1 out of 2 log speeds inserted. So what we did is to take those mice, breed, and then later on insert the second one. And the 2 other techniques on the top are the long single-stranded oligo and the DNA plasmid. And really what I would like to draw your attention to is the last part of the table on the in the rectangle in the correct log speed insertion. And as you can see, it was all significant. The efficiency in inserting both log speed was significantly higher for all the techniques except the 2 compared to the 2 guide, 2 oligo technique. What that means? It means that if I recap, it's all about the number of recombinations that are required for log speed insertion. So I explain what that means is basically when you insert the 2 guide, 2 oligo, so the log speed with the 2 guide, 2 oligo technique, you need 2 recombination events to occur. Whereas with the other technique, you only need 1, which is the insertion of the large chunk of DNA to be successful in generating the conditional allele. It doesn't mean that this 2 guide, 2 oligo approach doesn't work. It just means it's less efficient. And this technique could be very useful, for example, to insert log speed sites that are really far away from one to another. So what it means in terms of outcome for the two oligo technique. Firstly, we see a lot of one log speed site insertion, so that we can take advantage of this by generating the second one. And we see a lot of indels at the break, or the combination of indels and log speed, or log speed not in the right side, or the large relation between those exons, or nothing at all. So that gives a large range of outcome, and this is linked to the fact that this technique needs two recombination events instead of one. So what is my take on this? Firstly, as you've seen in this presentation, replication from a single laboratory is a waste of time, funds, and effort, and with little to no rewards. Importantly, and you probably have a vague idea now, post-publication peer review, social media, blogs, or preprint servers such as Biography is considerably changing the publication landscape. And to give you a very telling example how it has changed the landscape, let's give you an example. So in the 30th of May 2017, a paper was published on stating that CRISPR-Cas gives a lot of off-target effects in mouse embryo. I won't go into the details, but it was a landmark paper published in Nature Methods that day. And myself and colleagues were very much not impressed with the quality of the paper. There were some flaws we pointed out pretty much a couple of hours after it was published on Twitter. And this is, for example, my Twitter feed. And I decided a couple of days after to write a blog post about this paper and basically summarizing what are the flaws in this paper, why we shouldn't trust the data in this paper. And I did it 3 to 4 days after. So it was really quick. In a couple of hours, we find that the paper was flawed, we shared, and I decided to go with a blog post. What happened next is a little bit later, my colleagues published on Bioarchive server some concern about the papers. It's only 3, but there were many more on Bioarchive. It was a couple of weeks after. Way after this, pretty much a year after, the author published correction and a follow-up on this study showing that, in fact, the original study was flawed, which led to a recorrection of the papers over a year after. And this was followed up with well-performed studies, for example, showing from a colleague of the Sanger, showing that these off-target effects are not real, that was originally observed were not real. And we, with my colleague from the Sanger, we recently discussed this in this paper. So the last take I would like to give in a conclusion is we definitively need more multicentric, coordinated approaches to validate the technique. The recurrent problem of the CRISPR field and other fields in biology is too many studies have too small sample size, are underpowered, and what we've seen are in fact outliers rather than the general case. And this is really important to address, and with, I believe, the use of preprint, social media, multicentric study, we can address these issues. And I hope I have convinced that we can. So to finish, I would like to thank all the people in my laboratory that has been critical for this work, and all the funding agencies that helped me to perform the studies, and of course all the collaborators that are involved in this to guide to single-stranded oligo approach. And thank you for your attention.